Yes, thank you for the introduction, Christian. So my name is Elias, and I have a PhD degree now from KTH University in Sweden in the field of life cycle assessment, and I've applied that method to calculate climate impacts to biochar systems in Sweden here. But now I moved to another institution, the Swedish University of Agricultural Sciences, and I don't work with LCA anymore in that project, but I work with stability of biochar. And in the presentation today, I will go through first a presentation of our overall project, and then focus on the data and the modeling part of it. So first of all, why do we need to study biochar stability? All of you know that biochar systems contribute to climate change mitigation in various ways. Carbon storage, substitutions effects, and also field effects like reduction of greenhouse gas emission from soils in agriculture. But what LCA's results tell us is that carbon storage is actually the main and largest climate change mitigation benefit. So it's an important term in the climate equation for biochar. Second, it's also good to remind what IPCC published a few weeks ago is that biochar has a very large potential and high technology readiness level, and it is a rather affordable technique compared to other techniques. And you all know that it's already being implemented around the globe. On all the continents, we can identify biochar projects. But finally, we must also say that biochar can be produced from different biomass reactors, which then leads to different type of properties, and biochar can be used in many different environments. And that leads to a lot of variability in what we can expect from biochar stability. So biochar is stability is an important topic, and it's also very variable. But why do we need another study on biochar? Of course, many others have published on stability before. There have been reviews, but also quantitative studies and models to estimate stability. One of the first one was Biochar in 2010 with 34 observations. Then we had Budai and colleagues for the International Biochar Institute that had another study. And then we had a meta-analysis from Wong, and then Lehman in the IPCC published another one. And more recently, we had Wolf and colleagues who published another one. With a different number of observations over time and different amount of metadata. What I call metadata here is information about the experiment, about the incubation experiment. What do we know about it? So what was the composition of the char? What was the type of the biomass? What was the pyrolysis temperature? So what we can say from these studies is that we have seen an improvement over time on the knowledge we have and the models and their complexity. But still, there are major differences between the studies in terms of data sets, methods, and conclusion reached. And whenever we try to reproduce what has been published and want to check for data availability, we are not yet there. It's not that easy to do. And that pushed me to this claim here at the bottom to say that, well, there is margin for progress in terms of transparency of these kind of analysis. And this transparency is very much an important requirement when we want to design climate policies based on biochar. So, and that's why I'm very happy that we got funding from the Swedish Energy Agency to carry out this new project that we have called Biochar Stability, Supporting Transparent and Reliable Carbon Removal. We have 5 research objectives for our 4-year project. The first one is to actually increase knowledge on stability in Swedish agricultural soils with their very specific climate that has not been studied so much. We also want to establish long-term field trials, because you all know that these processes that are at play when we talk about biochar stability in soils happens over decades, and we should have try to have more experiments that are long-term oriented. And then we also want to create an open database for all the research data that there is available on stability, associated with new models, open source models, to make these calculations and treat and manage the data. And that will lead us hopefully to practical guidelines on how to use the models we have developed, both in policy as in carbon removal markets, carbon offsets, and so on. So the project is divided in 5 work packages. I can say that work package 2, 3, and 4 are mostly about the experiments. In work package 2, we are collecting different kind of commercially available biochars, and we characterize them so that then in work package 3 and 4, we can make laboratory and field incubations. The design of these experiments will start in this year, and then they will run for about 2 years, and then we'll analyze the results of these new incubations in Sweden and publish these results. But in the meantime, we also want to do some other work, and work package 1 is focused on the interaction between research and knowledge on biochar stability and the market users and the policymakers. So we'll have different roundtables and public consultations in that project. So if you're interested in that, register to our contact list, and then we will keep you in the loop for these events and discussion moments. And then there is a last work package at the bottom here, data analysis, where we want to do this, what I mentioned before, collect all the existing data and provide the code in an open source format so that it can be used for modeling. And we expect to have models already usable without our own incubation data, but with existing data. And then we'll update them at the end of the project when we can add the new data we've collected. The team behind this project is a bunch of researchers from the Swedish University of Agricultural Sciences. Cecilia and myself are there from the systems analysis side and the modeling part. Then we have Eric and Harald, who are from the, let's say, soil and molecular science side. They will be in charge of the incubations, and they will soon be joined by another postdoc researcher. The recruitment is ongoing to help carrying out these experiments. And last but not least, we also have Helena Söderqvist, a consultant involved in the project, to help us bridge the gap between market players and our knowledge. This being said, let's now dive into the work package 5, the data analysis. And there I want to present a bit about how are stability models built and what are we trying to do that is different from what has been done before. And then I'll go through a couple of preliminary results and insights. So biochar stability models, to create them, we have to start from measuring fluxes of carbon dioxide in the laboratory, as you have in this kind of machine here on the left. And then at the end, we need to correlate these, an estimated permanence or stability of the char to some easily measurable property, and we get some kind of an equation or a model. But to do that, there are many different steps. And so we have to start with first making experiments and taking a biomass sample, cultivating it. So we can choose different type of biomass, they have different properties. Then we have to produce a biochar from it, and then we can do that at lab scale, at commercial scale. We can choose different type of pyrolysis or gasification or HTC processes. And we have higher heat treatment temperature, residence time, heating rates as important variables here. Then usually we have to characterize the biochar, and characterization means here knowing the elemental composition, the heating value, and maybe other nice methods like chemical oxidation. We want to know as much as we would like to know about the biochar, and that needs to be characterized in the beginning of the experiment. And then we do what we call the lab incubation. So where we put the biochar in soil, we leave it there for a long time, several years, and we measure continuously over time, weekly, monthly, decomposition fluxes. Once we've done all that, we get what we call one controlled observation with also replicates, and that usually leads to a publication. So there are many researchers that have published these kind of studies. So when you have a couple of them, you can start compiling them and make a database. And once you have the database with all the data together, you can start doing the modeling. And the modeling has three main steps. The first one is what we call the extrapolation via curve fitting. So we have the experimental data and we try to look at what happens in the long term by using models. We call them exponential models usually, double decay models. Then once we have done that step, we go to the recalibration and the step and estimate calculation. So for instance, we can calculate BC100, the 100-year permanence, mean residence time, half-life, and apply different correction factors, for instance, to account for soil temperature changes. And then the last step is to actually then make a correlation attempt between the estimate of stability and the metadata we have from the experiment. So the biochar characterization, the type of production condition, and the environmental setup, and so on. And of course, this modeling is associated with many different choices that I try to exemplify here with some keywords of what is important. So my job in this project is to do the data compilation and to do the modeling as well. Here, these two colored boxes. What I can say about our specific objectives, the way we want to do it, is that we want to achieve basically transparency and reproducibility. So for that, we have defined some kind of guiding principles on what the data needs to have and how we need to report the data in our database, so that it really becomes transparent and that anyone can check the information easily in there. On the data analysis side, rather similar things. We also want the results to be reproducible. We want to exhibit, therefore, all the modeling choices we have to do in the process so that they can be discussed and exposed to criticism. We want to also use more advanced tools than what has been done so far, both descriptive and predictive analysis, and using models that are able to capture non-linearity. That's one important area of progress we can make. We also want to be better at trying to quantify uncertainties through propagation in the equations we are using. And finally, the code that we are developing, we want it to be well documented, and so that it's easy also to rerun the code in a couple of years whenever new data is available at low cost. So that's about our guiding principles, and which we believe will make our study a bit different. So now moving on to some results, the database structure. So whenever I speak about a database, I basically mean several large tables with numbers inside or with information inside. And these tables have to be well documented and well, transparent. So our database is made of 4 tables. The first one is called article, and it contains a list of all the scientific articles that are behind, that have performed biochar incubation experiments. And there we record when it was published, with the number of observations in it. We have a link to the publication. We know if, and we record these kind of similar information about every article. Then from the articles, we extract data, and that's contained in a table called this way. And there, the data refers to the time series, the actual measurements of CO2 released from biochar over time once it's placed in the soil. So we have decay rates, cumulative fluxes to record there, and we have collected data there for 121 individual observations, which is already more than what is available in the existing studies on biochar stability. And associated to this table, the second one that contains data is called metadata. And so in the metadata, we are collecting all the information about the experiments that was performed. Everything about the biomass was used, the pyrolysis conditions, the biochar properties, and the incubation conditions. And I highlighted before that the past models have been only collecting about 3 to 4 metadata fields. Here in this study, we really wanted to be exhaustive, and so we have 65 fields describing the experiments. And the full list is available actually online. I will give you the link a bit later. And finally, for the transparency, we have another table called validation, and that table contains two types of information, a location statement and a comment for every metadata field. So here an example is to say that, well, for the pyrolysis temperature that was reported for the first observation we have in the database, I'm reporting where is that information coming? Well, it's actually coming from table 1, page 23 in the original publication. And I also make a comment on the method, because these 350 degrees in that observation were not derived from a measurement, but were actually an assumption from the literature. So you see that this kind of validation information is really there to enhance transparency and highlight any assumption that was made either in the original publication or by the person who collected the data and put it in the database. We believe this is an important information to have and keep. So now that I've described the data base structure, let's look into it and see what we have collected so far. Starting with the biomass, we have in the database about 130 observations where we have metadata, and there we notice that most of the biochars that have been incubated are derived from wood. They really dominate the database, and then we have some crop residues and grasses also that are quite well represented, and these are, most of them are what we call C4 crops. They have a specific way of doing photosynthesis, which makes their use for incubation studies very easy because of some isotopic techniques that we like to use when we do these studies. Then when we look at the pyrolysis conditions, we can look at temperature, heating rate, and residence time. But there I'll just say a few things about the temperature. Well, the reactors were mostly slow pyrolysis and lab scale reactor, and the temperature was mostly in the range between 400 and 600 degrees. And we notice that we have a gap for everything that is above 700. We have nearly nothing, and we have only a few points for gasification chars at very high temperatures. That's a data gap we could maybe bridge when we do our biochar selection. And then in terms of biochar properties, if we just look now at the elemental ratios, carbon, oxygen, and hydrogen, we have a nice coverage of the space that could be reached, except for very low HC ratio. We know that commercially we have some biochars that have HC ratios of 0.1, but here we don't have any data, or not too much data in that area. We have mostly data for HC ratios between 0.4 and 0.7 there. Also worth mentioning is that, for instance, chemical oxidation test, which is used and presented also as a valid proxy for estimating biochar stability, is unfortunately not much used in incubation studies. So we can't really have yet make correlations or good correlations between estimated lab stability or incubated stability and chemical oxidation tests. Then when it comes to the incubation conditions, I can say that we have mostly laboratory incubations and only 8 field observations. The longest experiments we have in the database is 8.5 years, but most of the studies are between 1 and 3 years, let's say. Soil temperature in the incubation was most commonly between 20 and 35 degrees and up to 40 and 60 degrees in some cases. In lab conditions, you can do that mainly to accelerate the decomposition and see if temperature plays a big role or not. And temperatures below 15 degrees, what we have in the database are a few points. They are mostly the field studies, so not lab, but the field. And there, in that case, the temperature reported is an annual average, although it varies day by day, season by season. Soil types, we have quite a different type of soils covered here in the data, but I want to highlight the fact that the moisture in the soil is usually kept constant, around 60 to 70% of the water holding capacity. But we have also 5 observations that are modeling flooded conditions, so with 100% moisture there. And if we look at origin of these experiments and the soils that have been used, we have a lot of data points from Australia. few studies in China, some in Europe, and quite just a few ones in the US. But we can also note that there are no incubation studies performed in an African context or even in an Eastern Asian, Middle Eastern scope. Then last about the data we have compiled here is this. This is actually the most important data. That's actual measurements that have been done of how biochar decomposes during the experimental time. I like to show it because not many persons have actually seen the data. And I also want to thank all the authors who have shared with me their Excel files and other kind of data formats that I could then retrieve and compile, because it's a very, very time-consuming process to collect this information from PDF and published articles. So moving on to the data analysis part now, so the second part of my work. I'm just going to describe briefly the status of the implementation, because we're not done with this part. And I will start from the last step. So in the correlation to metadata, we have developed code to be able to reproduce the simple analysis that have been done before in terms of linear regressions. So comparing the 100-year stability to HC ratio, highest treatment temperature, and organic oxygen to carbon ratio. But we also want to try multivariate regressions and decision trees or random forests to try to find better relationships in the data we have. And that is enabled by the fact that we have 65 metadata about each incubation. And we want to calculate propagated uncertainty there. That still needs to be done. When we talk about recalibration steps and estimate calculation, we have implemented what we call the temperature correction using the Q10 method. That's commonly done in the field, and we also are going to do it, but we're also investigating whether we could do moisture recalibrations and field data corrections. In terms of estimate calculation, we're going to, of course, calculate the classic ones like the 100-year stability, the half-life, the mean residence time, and we could also do the 500-year stability, for instance. But every time we calculate estimates of stability like this, we actually lose some information, and we are considering to work with other kinetic estimates that don't induce information loss. And last but not least, the first step is extrapolation via curve fitting. It's actually the most crucial step in the modeling, and that's where we are putting most efforts now to get a good work done. And that will lead us to implement physical sanity checks about the fits we get, assess the goodness of the fitting processes, detect and avoid overfitting, and also detect whenever the model outcome is uncertain because of what we call non-uniqueness. This is quite mathematical challenges with curve fitting, and I will explain, maybe show a bit more how it looks like in practice. So the curve fitting step basically means that from the experimental points, which are these crosses here, that's one observation, what has been measured in the lab. We want to try to find what happens in the long term, and we use these kind of double exponential decay models, these curves. And trying to fit and find the best curve for that is a challenging mathematical problem, because we evolve in non-linear spaces. And to be sure that we have found a good fit, we always need to do this visual inspection. We must not just run the code and get the numbers out. We also must look at the figures manually. And then the first step, we also need to check whether the different models lead to any significant differences in a 100-year perspective. There is something we call the residual plots. So the non-explained variance or the error we have in the curve fitting step, it should have a random pattern when we have a good fit. So that we need also to check. And then we can do more advanced things like looking at the covariance matrix, calculating different kind of goodness of fit indicators that can help us detect overfitting situations. And when we do all these things properly, we can get to a best fit that we can use. We can calculate its uncertainty and also demonstrate the uniqueness of the solution by plotting these kind of weird figures here. I'm approaching to the end. I'm just going to show this figure here that I think we'll try to illustrate why this curve fitting step is really important. So this is the data, the 100-year stability that is reported in the latest stability study by Wolf and colleagues, published in September last year. So you see different observations that they have in their data set and different stabilities. And my question is here is, am I able to reproduce these calculations, knowing we have the same data as information? And I did that 2 days ago. So it's very preliminary results. And you see, I did some very naive curve fitting steps. And you will notice that indeed, yes, for most observations, I can obtain the same results as him, as in this other study. And that you see when you look at here, the fact that you have a red cross and a blue dot on top of it. So it happens for quite a few cases. We can obtain the same results. But you also see that if I apply different fitting strategies, different algorithms, different modeling choices, I can get very different outcomes. So that is something we have to bear in mind, that we need to have the right fitting strategy. And then you also see that in other cases, we have, with these naive fits quickly performed, I don't necessarily get what has been published before. So now I need to understand why didn't I get the thing and what is very specific in the modeling steps so that we get the same results, or was there a mistake at all? So this is for me a demonstration that fitting strategies and the modeling choices have an importance here, and they need to be reported also when we present our results. That is work to be continued. And so now that I've said all this, maybe I scared some people regarding biochar stability, because I'm opening many boxes and going into the details of the modeling, but I want to clarify here with what we know about biochar stability. The first thing we know is that there is not a single stability value for all biochars and all environments. There is variation. But what we do know also is that biochar stability is much, or the biochar permanence in the environment is much longer than the raw biomass that you had in the order. Decay rates are at least 1 to 2 orders of magnitude smaller. And then finally, last but not least, we know that stability increases with higher pyrolysis temperatures and with lower HC and OC ratios. For us in the research project, the next step will be to continue this modeling. We want stability modeling to be transparent, reproducible, and additive. That's really what we try to achieve here. And we are building for that the largest biochar incubation database. We want it to be open source, and we will also provide all the code that is needed to analyze it. Thank you for listening. And if you have any questions, we can answer them now.
Thank you, Elias, for these insights into one of the main characteristics of biochar. So just a quick, we are open for questions. So if you have any questions, write them in the chat or unmute yourself and ask directly to Elias. My first question would be, so if I assume you know the IPCC assumptions and also the CDR market assumptions about biochar's carbon stability. Do you think they are far off, or are these relatively conservative values almost okay?
Okay, thanks for the question, Christian. First remark I want to say is that when we do a model like this one, we get a relationship that can be used, but the raw model can also be modified and be made a bit more conservative for use in specific policy context. That's also one remark I want to make, that the research output can be slightly modified to be more, let's say, to account for more uncertainty, and then use in policy as such. Then you talked about the IPCC model, right? And also the models from that are used in carbon removal markets. So I think there are some actors that still use one value for all biochars. I think that's a bit limited today. There are others that are using models that are derived from science. And so the latest model available by Wolf and Lehmann and colleagues from September 2021 is also being used in the carbon market. So that's really nice that science gets used in a good way. And the IPCC model that you mentioned for the 2019, that is an appendix to the greenhouse gas inventory guidelines. I don't know if it's being used because it ended up in an appendix of that report, and now it has been updated by the authors themselves. So we should probably use instead the Wolf paper rather than the Lehmann 2019/IPCC table. Did I answer your question, Christian?
Yeah. You also got a question from Trevor. Do we have any useful data from ancient carbons that can say anything about stability in your work?
Yes, that's also something that has been, for instance, in the earlier work from Spocast 2010. We know that there is pyrogenic carbon that is spread in the soil in various locations, and we can still study its decomposition today. I haven't seen decay rates about these recently, but I think it's interesting to look into that. One of the challenge with it, if we can't use it in the same way as when we do incubation experiments, because we don't know the initial quantity of carbon that was there. We only know how much is there today, but we don't know how much was there initially. And then we can look at the decay rates that they have today, but we don't really know the initial quantity that was there. So we can't do the same models, but the decay rates can be interesting in themselves. When we can compare the decay rates from old biochar or aged biochar or ancient biochar, there are different terms with slight difference in meaning. And then compared to the decay rates we measure with fresh biochar today. Yeah, that's an interesting thing to do, I believe. We haven't done it yet, but I will take it in and look at decay rates, not just the curves, but the decay rates. Yeah. Thanks, Trevor.
Another question from Kathleen. Do you know if the other CDR solutions have done or are doing similar exhaustive research on durability?
And am I also looking for non-soil uses?
Yes.
So, other CDR techniques, if we talk about geological storage of CO2, BEC, CCS, and all that, they do, and I think there is no debate. They have a very much longer permanence than biochar would have, and similar techniques. In the field of, let's say, enhanced weathering, where you apply some minerals to soil, I know that they are doing modeling as well to try to determine, carbon removal quantities to quantify that carbon stored. But I haven't seen yet a very well working model for that, but I think it's in progress. So yes and no, I would say to Kathleen that some need to rely on modeling, others have done the modeling and it's very easy, or it's easier in their case, like CCS. Yeah. And am I looking at durability of biochar in non-soil uses? Unfortunately, the project is only looking at agricultural soils. We would have been interested by urban soils as well, where biochar is used, for instance, in biochar macadam mixes, as it's very well commonly done in Stockholm. But use in asphalt and concrete are not yet on the research agenda. The durability of these. But I'm very interested in these topics, and I've been talking to some actors that develop these products to see what they think about durability there. But it's an important question as well.